to recall and refresh you from the previous lecture. I hope you all remember the story when we talked about the rise and falls of the stars. We talked about some Nobel Prize winners, Dr. John B. Gurdon and Shinya Yamanaka, who received the Nobel Prize for their contributions of doing some very original work in the cell reprogramming area. Then we also had some really fraudulent research outputs from Dr. Haruko Obokata and Dr. Wang Woo-suk, who did various kind of fraudulent studies, and you have seen the fate of those studies as well. So today, what we are going to talk about various step, rated study which we have seen last time, stimulus triggered activation of the pluripotency. So what happened to these scientists? I just want to give you a little bit idea that after the study was taken away, retracted, what happened to the scientists who worked in this particular study. Then we'll talk about some of the scientific papers, retractions, why it is happening, and then we will have some discussion about research misconduct. So let me give you some idea that what happened to these staff scientists, Dr. Haruko Obokata and others, after the paper was retracted. So Dr. Obokata, she agreed that both her research papers published in Nature should be retracted because the study was not conclusive and having several issues. Nature Journal confirmed the retraction on 2nd July. And Obokata was actually again allowed to join Riken Center, given a chance to replicate her own study. But she was kept under the CCTV video surveillance to replicate her study. But after 6 months of this tension, in December, she was failed to reproduce her staff cell work, showing the stimulus-triggered activation of pluripotency. And then she resigned from the Riken Center. Further to that, Riken Center considered having some criminal charges against her for misusing the research funds. One of the co-authors of the paper, Masatoshi Takeuchi, returned 3 months of the salary. Hitoshi Niwa was given, disciplinary warning. Chiriyoko Wakayama, another researcher, was given suspension for up to a year. And a former, senior scientist, Dr. Ishikawa, mentioned that some of the embryonic stem cells what Dr. Obokata used were actually stolen from another research labs, which actually warrants the criminal charges. So based on these, it was really unfortunate to see all the authors, co-authors, everybody got penalized. But what was the most disheartening to see this particular clip, that what happens to one of the co-author, Dr. Yoshiki Sasai. Please watch this clip.
The co-author of an infamous and controversial stem cell paper that was eventually retracted as die of an apparent suicide, Yoshiki Sasai, heralded as the brain maker by the journal Nature in 2012, was found dead Tuesday in his research building. Sasai co-authored a stem cell paper released in January that claimed stem cells could be created using a new revolutionary method called STAC, or Stimulus-Triggered Acquisition and Pluripotency. The widely reported technique was too good to be true, and the paper was retracted in July after attempts to replicate the study failed and other researchers found signs of plagiarism and misconduct. While Sasai was found not to have committed misconduct himself, the lead author and his protege, Haruko Obokata, was eventually found guilty of fabricating parts of the study. He issued an apology in early July saying he was deeply ashamed the research had to be retracted and that he didn't catch the errors. As to whether the retractions and surrounding media attention played any role in his death, a spokesman for his lab told the Washington Post, "It's certainly possible, but I don't know what else was going on in his life, so it's very hard to speculate. He had the stress from the retractions, and there was also the prospect that he would face disciplinary action due to the finding of research misconduct." According to NHK, a Japanese broadcasting corporation, 3 letters were found inside a bag at the scene. One was addressed to a Center for Developmental Biology official, another to a lab member, and a third to Obokata. Sasai's loss was mourned by scientists around the world, with a stem cell scientist in Boston telling the Boston Globe his death is a huge loss, and that he was a gifted scientist destined to continue to make momentous contributions. And a Canadian doctor in Toronto told Nature that Sasai was a rigorous and innovative scientist, and his loss will be deeply felt. Obokata is currently attempting to replicate the stem cell STAP findings while under video surveillance at the Rican lab. For Newsy, I'm Jay Struberg.
All right, so this was very unfortunate to see. He was not the lead author of the paper, but somehow he felt ethically very much responsible for this whole issues and committed suicide. So you have seen what all level of penalties and issues have happened after publication, after retraction, and post retraction. This was not the only paper, not the only study which actually happened in this way. So let me also show you a couple of more examples where scientific papers have been retracted, which will also help us to analyze what are the possible reasons for these kind of scientific paper retractions. Let me show you a couple of papers as an example. So this paper was published in Blood journal, which is a very prestigious journal, and the authors later on were found guilty of showing some duplication of images and multiple figures having irregularities, and the paper was finally retracted. Another paper which they studied about FOXO1A, which acts as selective tumor suppressor in the rhabdoma sarcoma. It actually led to the not only a decent paper in Journal of Cell Biology, but also commentary in the PNAS. And again, due to the irregularities in the paper, it was retracted. Another paper, another group was, in fact, really ambitious. They used this particular gel image in one paper and same image in another paper from the same group, and that was in the Blood journal. So now these papers, I think both papers got retracted. Additionally, there was the news in Nature that, a scientist, a crystallographer, faked almost a dozen structures, protein structure, which he has really reported, and unfortunately turned out to be one of the Indian origin scientists at University of Alabama, Dr. H.M. Krishnamurthy, who was involved in almost 10 or so papers which are published in very prestigious journals like JBC and Active Crystallography. You can keep looking at Journal of Molecular Biology, Cell, Biochemistry, PNAS, Biochemistry, PNAS, Nature. So again, all of these are very reputed journals which are going through very rigorous peer review process. But after that also, this thing happened and so many papers got retracted. In addition, this paper, which was looking at the antimicrobials from the plant root, Arabidopsis thaliana, this paper got retracted. But the reason I'm showing this paper is not only that this particular paper got retracted, it also led to the discussion that, whether these kind of papers which are getting retracted, what happens to the other papers which are citing these papers as a key paper. By the way, am I audible clearly, guys, or any issues in looking at my slide and my word? Going okay?
Yes, sir, it's pretty audible.
All right. Thank you. So this particular paper was not only, let me go back. Yeah, it was not only retracted, but also it led to the discussion that if the same paper, same work is also discussed in any other paper, then what happened to that paper? So for example, this paper was also a key reference for other paper, Journal of Agriculture and Food Chemistry, and several of the authors actually of the same paper were listed as authors in the other paper also. So then ideally one should retract even those papers. So this is kind of a really tough situation if these things happens. Let's hear about the publisher side. Elsevier executive publisher, Dr. Jaap van Halten, his views about how some of these kind of fraudulent studies comes into the light.
When it comes to research or scientific misconduct, then it is not possible to compare a manuscript, a faked, fabricated manuscript, with something that has been published in the past, for the very simple reason that it's not copying, but it is a generation of new data. Most of such cases come to our attention through whistleblowers. And these whistleblowers can come from everywhere. But it surprises me, it strikes me even, that most of these whistleblowers live quite close to the fraudulent author. Often they're colleagues. These colleagues, they send us a message and say, well, can you please have a closer look at this paper? Because the raw data don't exist, for instance. And then we dig deeper into this, and if there is something really wrong, then we will find out. But we often need those external people who come to us and point us at a case of misconduct.
All right. So most of the time, of course, if you are doing this kind of work with the colleagues and with a group, people who are part of it, many of them reveal this kind of misconduct, which is actually really important to inform, so that this kind of activities cannot be promoted. So the question is, what are the possible reasons for the retraction of scientific papers? And even I did a PubMed search, just a few days ago, and I could see that if I use the search words retracted articles, it identified close to 2,000 articles which were retracted just in last 1 year. And of course, if you go back, it will know the papers started getting retracted from 1970s or so. So there is a large number of papers which are getting retracted by the journals for multiple reasons. And the reports from the Office of Research Integrity, which is ORI, makes these kind of retraction announcements in scientific journals, and also analyze the reasons that why these kind of papers are being retracted. So journal PNAS, which is one of the prestigious journal, Proceeding of National Academy of Science in USA, they published a paper earlier that misconduct accounts for the majority of retracted scientific publications. And after the analysis, what they showed that if you can look into these 4 colors, the fraud cases in the red, errors in the blue, plagiarism in the yellow, and duplicate publication in the green. So large number of and as the years are going, you can see there are more and more publications are getting retracted. So the peer review process, so as the journals have now become much more alert that these issues might happening, so they are really being more stringent now. And over the years now, the process is becoming much more stringent to make sure these things can be kept under control. But despite that, you can see that their numbers are rising. It means so many people are able to still go to the next steps of publishing the papers, and then later on the papers are getting retracted. So the fraud case, as you can see, is close to 470 or so numbers, and then next is the error based and plagiarism based. But also there are many duplicate publications, which is very much worrisome, that how people can publish the same data in 2 different journals, 2 different papers. And the years of retraction, if you see, is really rising. So when they analyze the data, they found that only 21% of retractions were because of any errors made by the authors, but actually majority of those, almost 67%, were because of the fraudulent case, and 11% were incomplete or uninformative or having some issues. So, but one thing which comes to the journal's report, that most of the papers which gets retracted are because of some or other issues of the image manipulation. And I think briefly we discussed earlier as well in Dr. Obokata's case in some of her papers. But likewise, there are many papers where authors have tried to play with the images and try to show the way they want to interpret the data. So like in this case published in Journal of Cell Biology, the original image looks like this. If you look at lane 1, 2, 3, 4, lane 3 has this particular extra band, but now in this particular image, lane 3 is having this particular band made very faint and somehow got disappeared. Now if you look at the panel B, 1, 2, 3, 4 lanes. And now again you look at the manipulated image, 1, 2, 3, 4 lanes. So you can see this particular thick band has appeared new, whereas this particular band is totally disappeared here. So it's very interesting how people are able to come up with these kind of images, and totally they are able to add and remove the bands and the which will totally change the entire result. Whether you are able to amplify a right band or you are able to get the contaminations or what is happening over there, it's not clear. So this is one of the case when they are showing the gross manipulation of the blots. Adjustment of brightness and contrast it happens very often. So the original image looks like this, and the image which is manipulated looks like this. So again, now these bands are totally getting fainted. If you look at again adjustment of brightness and contrast, after sequential brightness contrast, now this particular band is probably not looking totally clean protein. So if you are purifying a protein which is having a lot of contamination, which of course you should not use for any enzymatic assay or any kind of experimental work. But in this case now, if the author want to show that they have got a pure protein, just by adjusting the brightness and contrast, they are able to reach out to get a pure protein. But experimentally, it takes a lot of time and effort and so much troubleshooting to try out multiple ways of chromatography to get a pure band, right? So it's such a neat band which usually very difficult to get in the experimental conditions. Again, another image, original and manipulated image. You look at multiple bands here, could be degradation product of this protein, and here it is totally clean. So as the next particular band. So a lot is happening, right, in the background cleaning and image manipulation with the brightness and contrast. In this case, looking at some electron microscopic images, this is the original image, which is showing here, and now the image is manipulated. Is showing a lot of these dark particles. Maybe they're looking nanoparticles, and now they want to show the enhancement of this feature. Additionally, these are the images shown from a microscopic image from the paper. Now this particular part of the cell, now when they try to check is that the real good image or not, it looks like the image was actually like this, and it was somehow changed and manipulated like this. So a lot is happening by playing with the images, by changing, and then interpretation is the way you want to interpret the data. So science journals, they really now crack down on the image manipulation. They have even their in-house editorial team who looks for a lot of not only plagiarism checks, but also the image check before it sends for the peer reviews. It was interesting to read whether now we need some digital detectives. And so one of the computer scientists and applied mathematician, he ended up with an interesting job, which is to check the digital images for the journals, and his role become like for FBI, do digital detective. So again, we need this kind of individuals and maybe more trained individuals who can check the quality of these images for the journals to make sure that data quality has not been changed. But it sometimes becomes very thin boundary of what is acceptable, what is unacceptable, right? I'm sure when you are clicking a picture and you want to send to some people, you always play with a little bit, how to make it look better. Now, what is acceptable in that, and how to still not avoid any kind, or how to not infringe into any kind of plagiarism issues. So, there was a post earlier made by Dr. Anurag Chaurasia. He mentions that my personal experience says research supervisors and guide also need to be trained to detect these manipulated images, sometimes being submitted in thesis by the students. So I think that's pretty valid, because many times the mentors, the supervisors, the guides, they are more senior people, and the students are much younger crowd. So you have much idea probably for the latest softwares and editing tools as compared to your supervisors and guides. So even the guides and supervisor and the senior investigators, they also need to be trained to make sure that they can understand that this image which comes to for a paper review is actually right image with the raw data quality, or is that something being changed by the student? So as I was discussing that, what is acceptable practice, everybody want to show a good effective figure for the presentations. And when you see the good papers coming out in journals like Cell, Nature, Science, you do see the quality of the figures, they look very pretty. And many times you wonder that, okay, how these kind of, authors have made these figures, they look so good, and I should also try to make my figures look good, right? Just to give you the a little bit feedback about how these journals work. So prestige journal like Nature, Science, Cell. Sometime when we have being associated with these journals for different papers, the experience has been very different, right? So these people, they look at your idea, your data, but then they help you to present it differently and very effectively. So once they have accepted the paper, they also take it their responsibility to make it really look effective and how the whole thing should be really presented. So they will give you more guidelines and make the things look improved. You don't have to try to change your data and figure just because you want to show that kind of quality to these journals. So it's really important that while you are making any changes in the figure, of course you want to show to look at much more clear. So if you're changing the contrast quality or brightness quality, you can put those settings detail what you have changed from the original figure in the legend. And also you can provide the raw image in the supplementary. That will definitely rule out any possibility that you have tried to change something. Even if without knowingly you have somehow made too much extra look, which looks now the things different. But as long as you have mentioned those settings and you have given the raw data, then it means your intention was not wrong. Even if an reviewer might conceive it differently, but you will not have any blame for that, because you have put the raw images and raw data directly available to them. So I hope this brings to the major part of today's part, which I really wanted to have discussion about the research misconduct. I hope by now, from my previous lecture and today's some of these papers, I have made a strong point that there are a lot of research misconduct happening, and people are presenting different type of images, different type of data, even duplicate papers. Variety of issues have happened, and different papers have been retracted, so as different authors have also got a lot of penalties. So let's discuss the third point. But just to brief what is considered the research misconduct, that is fabricating data, falsifying data, or plagiarizing data in proposing, performing, or reviewing research, or in reporting research results. So as per the National Science Foundation in US, the fabrication is essentially you are making up the results and reporting them. Falsification is you are manipulating the research material, equipment, or processes as per your requirement of the objectives. And plagiarism is you are taking somebody else's ideas, processes, results, and not giving them proper credit. I think we discussed that in the first class as well. And I'm really going to be looking for all of your write-ups from the assignment in this slide, whether you have tried to write originally, and how much plagiarism check we will do that to see the percentage similarities, and also whether if at all you are using any reference, are you giving credit to the original authors of that work. So let's hear one of the experts from Elsevier, Dr. Garrett Tibbs. What is research misconduct?
Scientific misconduct, or the fabrication and falsification of the scientific record, and the act of plagiarism in conducting or reporting research, is really the most corrosive transgression that one can make in science. Because while the self-correcting nature of science means that incorrect data entries will be pruned out from the scientific record, the damage done to lives and careers, the wasted research efforts cannot be changed. That will persist. But even those conditions may be, those problems may be not the most serious result of scientific misconduct. Death has resulted in clinical trials from scientific misconduct. So if that's the nature of scientific misconduct, there is a clear need to act if you perceive that you've witnessed scientific misconduct.
All right. So again, research could be of so much paramount significance that, many times clinical trials are going on, and think about right now, there is so much issues for COVID, right? Whether a drug is working or not working, whether this particular vaccine is safe for, giving to the patients. All of these goes through some research work, and a lot of research data is being generated. All of a sudden, you have heard that, there was, one of the Ayurveda medicine came all of a sudden with the claim that it can cure COVID, right? I will not name it, but I'm sure you understand. So then, all of a sudden, it looked like, somebody found a cure, but when they looked for what is the scientific rationale of it, it was found that the study was very biased. They tested their Ayurvedic, drug product on the individuals which are healthy young individuals who have had COVID mild symptoms and have recovered. So now, if you are giving the drug to the young individuals, they are going to cure anyways, right? Because they are not going to get too much infected with COVID infection. But when you are actually giving them even supplement, that is probably going to help them better to recover. So the study was on a very small number of sample size, and also done on the very biased population, which was chosen to prove the point. And then, of course, then the company and the leaders of that product, they backed off, and then they said it can be used for the health supplement, which of course can be given easily. So, but it was at least taken back, not as the drug and cure for the COVID infection. So again, sometimes these kind of misleading results and findings could be so much alarming and could have so much detrimental impact on society. So coming to the scientific misconduct types, so there could be multiple ways, major types. One is the publishing misconduct, where even the data may be okay, but even to acquire data, sometimes the practices for collecting samples or doing the clinical trials, even they were not ethical. Or of course, research misconduct, that's what we are talking more. That is essentially data is fraudulent and having a lot of serious issues in the data. So let's see the view of another scientist, Dr. Dana Fisher, that how to maintain highest research standards.
Their obligation as researchers to hold up their colleagues to the same level of standard that they hold themselves to, and that we all need to hold ourselves to. And I think that's particularly important when there is such a risk, particularly because if there is research misconduct taking place at an institution where you're affiliated or where you're housed, and you don't say anything about it, it lessens the quality of the research overall. And in the end, it will likely lessen the quality and the perceptions of the quality of all research coming out of that institution. But I mean, I think it's not just because other people might perceive the research as being bad that one needs to report it. I think it's also that it cheapens everybody's research. And I think that it is really important that we hold ourselves up to the highest ethical standards and the highest research standards with the way we conduct ourselves, with the way we deal with research subjects, with the way that we deal with colleagues or research assistants who we're working with. And in all those cases, it's really important that the conduct be befitting of a researcher. And otherwise, the people who are working underneath us won't learn the ways in which we need to conduct research. And it could be just, a downward spiral.
All right. So, as Dr. Dana Fisher mentioned, even when I visited, Seoul National University, so still, that dent is still there. That kind of damage is still being made, and people still, recall the issues of Dr. Wang Woo-suk study and how that has really given them setback in the entire area of pluripotent stem cell research. So when this particular journal PNAS, they analyzed, what kind of situation happening for research misconduct. This data is slightly old, but at least it gives a trend. I think trend will not change, but this is not the most recent pie charts. So what they looked at, the kind of fraud or suspected fraud papers, and the blue color is the USA, the largest numbers, followed by Germany, Japan, then China, UK, India, South Korea, and then others. So I'm sure you'll be very happy to see India's very small fraction here, and must be wondering why USA fraction is so large, right? I don't think it represents the the cases appropriately, because USA does a lot of research. There is a large number of publications come out, and in USA all populations are there. People from all over world, they're working as researchers over there. So of course number of papers are high, number of big groups are big, ambitious researchers are there from all over the world, and that actually ends up with the large number of these kind of fraud papers. Of course, the the readership is also quite vigilant over there. Again, I have shown you one of the student. He was reading some paper and sent an email to me that what paper you wrote in 2008 is copied by somebody else in 2011, exactly same paper. How is it possible? So again, when people are reading, they're also very critical about it, and then they report these things. They don't just read and just hide that. So that's why USA is much larger proportion, so as the Germany. Coming to the plagiarism checks, again, USA stands quite tall. Other countries share their pie. India definitely shows here much larger, fraction. Duplicate publication also India shows large fraction, because I don't know what all possible reasons could be there, but plagiarism is definitely a case. We are not the English-speaking country. So people, when they are writing, then there is a general tendency to do copy paste rather than thinking about writing on their own, and that do lead into serious problems like this. So plagiarism is acceptable in that way, that why India has shown so large proportion. Not acceptable, plagiarism not acceptable, but why India is showing the large proportion, that's what is acceptable here. But this part I'm not sure why India is showing so many duplicate publications as well. But nevertheless, to see your country in these kind of pie charts itself is alarming. It's not a very good thing to see, right? And when they analyzed even the impact factor, does it make any difference? That what kind of impact factor papers are showing the errors or plagiarism or frauds? Looks like most papers are in the the small impact factor level. As the impact factors are keep going further, number of retractions have been less. But usually the high profile studies and really breakthrough studies which is published in the big journals have been quite a few which have been retracted. So when people have looked at the journals with the most retracted articles, in this case the Science had almost 70 articles with a very high impact factor. PNAS 69 articles, JBC 54, Nature 44. So you can see all these top journals have large number of retractions. And again, reason is that on one hand, when you publish something of that caliber, it is being read by large number of people. When you publish something in Nature or Science, large number of people are going to analyze your data, looking at replicating your study, and then sometimes they find these problems. Then they put the words to the editors or reviewers, and then again when people are not able to reproduce the results, then that comes into the serious problems of these issues of retractions. So it just gives you the feel that even not the big journals are also not 100% proof. Even they have so many issues like this. When the reasons for these kind of retractions were analyzed, although the small number of papers which were looked at, so fraud cases were quite large. Suspected frauds 192 in this case, plagiarism 200, duplicate publications are also quite large. Errors are still significant fraction, and some of these are other and unknown. So I don't know, how people are able to, publish the same work in different journal. That's very alarming, right? Because when you are, submitting a new paper to a journal, ideally journal will do the background search of this work, not having similarity with the anybody else's work, and if author's own work, then they should definitely not be considering for publication. But looks like many papers makes through, and that's where a lot of low impact journals, and many times even journals which are not having even the proper indexing, they take any kind of paper. Every day we get almost 10-15 email for manuscript submissions from so many open access journals who just want you to submit anything, and for the sake of filling their catalogs. Of course, they charge even the pay charges for the payments for these papers. So that probably leads to all kind of reducing the qualities and these kind of papers. And as I mentioned that most of the time when a big journal publishes a study, it has very large impact. So like Lancet published a study in 1998 which has almost 758 citations. So it means people have really 758 people found this study to be so significant, they made citation in their own research. And the study was actually retracted, one of those was 2004, other was 2010. And likewise in the studies published in Blood, Science, Lancet, Science, Nature, Medicine, so many citations, 700 and 600, all these large number of citations, then finally came out to be the study was fraud or error. So all of these are really big problems, big issues. So that leads us to some discussion point. And first point being that what are the probable causes of research misconduct? So let me now take some of your viewpoint on this, that what you think could be the reasons for having these kind of issues. So please open up and make it interactive, give your perspective. Why you think these kind of research misconducts have happened, and what lead these authors to do these kind of mistakes? Anybody want to add their points? I hope you're all not sleeping. You're not all afraid too much from looking at these slides.
Good morning, sir.
Hi, good morning.
Sir, I feel that basically people are really greedy about being famous. Like maybe that drives them to copy some other people's study to become famous and all that.
Okay, I think that's a very candid comment. Anybody else want to add some more points?
Good morning, sir.
Good morning.
Sir, I think they may have some time constraint, like they may have some deadline, so they want to finish it before that time, so they manipulate this data.
Okay, other comments?
So basically, they try to get a shortest route to success, so that's why they perform the fraudulent research. So that could be a main reason.
Okay, some good points are coming.
Yeah, good afternoon, sir. I think sometimes it's about relevance as well. Since you're in science, so you have to publish to stay relevant in the scenario.
Mm-hmm. Okay, I think good points being made. Let's have some more comments.
Yeah, hello, sir. It's many times there's a lack of original thinking, and these things make people to copy the points from others and then publishing as their own work. So lack of thinking and lack in their mental process.
Definitely valid point. What else? Yes.
Okay, sir. I will come back in a minute.
Sure.
Yes. Some researchers might not get proper result. So, what they do, they just took the result or data from another research.
Right. But the reason is why they are doing that. So the question is why this kind of research misconduct is happening.
So, in order to further get more government funds, like using this data, you can present and get more funds for the upcoming projects.
All right, good. So I think it is all great points. All of you have really covered the things very well. Starting from looking at to become famous, to get more grants, to get more money, to get publications fast, to meet the some of the quick deadlines. I think several of the points which all of you have mentioned are very relevant. And I think that
Yeah,
pretty
and
yes.
also be relevant in what you can say current trends as such, maybe.
So correct. So I think what you're adding is in the same light that you need to publish fast. You need to show that you are able to be on top of that field. And so based on these, I think these are the same point which even I thought and to be summarized. Uh, Many times, one thing is really crucial to mention here that let's say you are supposed to submit your thesis, right? And university has given deadline that or made a guideline that you have to publish 2 papers before you graduate, and now you have only 1 paper out of your 4.5 years of work. You have left with only 6 months time, and that time there's a lot of pressure, right? You are really under tremendous pressure, and you want to see that, okay, can you publish 1 more paper in that time? Or somebody is having a job as an assistant professor and want to get promotion as associate professor, and during that time they have already published only 3 papers, but they need 4 papers for the promotion to happen. So they are also under pressure, right? And likewise, people are at different level of career pressure. That is one of the major reason why sometimes these kind of things happen. I think many points which we discussed were all relevant, but career pressure is one of the most important part. Funding problem, I think some of you have already stated. Hunger for reputation was already started in the beginning. And also, as some of you mentioned, that it's easy to get data from somebody and fake them rather than trying to think originally, which is very difficult, and then trying to do original work. So these are the probably causes for research misconduct. But I hope this discussion is also leading you to think about various these points. We don't feel to reach to career pressure at any stage, and think about various of these points which should not affect you. So let's uh, listen, Dr. Dana Fisher again, that what steps to be taken if you are suspicious that there is a research fraudulent is happening.
The steps that I think should be taken when one perceives research misconduct, in all cases, I think that the person who is perceived as doing the research misconduct should be addressed first. And in some cases, a person may feel uncomfortable about doing that. But if the person is somebody you feel comfortable talking to, I think it's very important that you go and speak to them first. It's a very bad idea to come out to the university, to the department, or even to the public regarding a perception of research misconduct. However, in some cases, people might feel uncomfortable with it. I think that in a collegial manner, if it's possible to say, I'd like to discuss this research project you're working on, or X, Y, and Z that I feel that you may have done in a very open-ended way would probably be the easiest way to discuss it. If, however, somebody feels uncomfortable approaching this person for whatever reason, be they a senior colleague, somebody who's going to review them for tenure, somebody who hasn't ever liked them, or somebody who's their advisor or has certain requirements of them as a senior advisor or professor, then it may be more comfortable for a person to go to another person in the department or somebody who's seen as an ally. And I think that it's very important to identify who would be a comfortable ally to discuss your perceptions regarding research misconduct with, and then to speak to them in an open-ended and non-accusational way about the specifics of what you perceive to be happening. And then hopefully that person would be on a more equal level of power to discuss either with the colleague or to discuss in a more open environment the research misconduct. I would say that in no cases should anybody ever go forward with their perceptions about misconduct without giving a person an opportunity to discuss if it is rather a misperception or particular aspects of the way they're doing their research in a private way that's not transparent, that makes it seem like there's research misconduct taking place, but there is in fact not.
All right, this leads to our next discussion point, that if you are suspicious that some research misconduct is happening, then what you should do. So let me again come back to you, and let's take this point that let's say you're working in a lab and you are, let's say, senior person, right? Like, you are 4th-year student, and you see there is a junior colleague who have started working recently, but when they show the data in the lab meetings, looks very neat data, and lot of things are not documented properly, and the results are actually not accurate in your mind. Second situation could be that you are a junior person who started working, and you see that a senior is actually doing the things which you feel it is not correct, and you are not comfortable with their data. So now these are two different situations, right? And now what if you are suspicious that there is something wrong going on, and then how you would like to address that kind of situation? So Dr. Dana Fisher mentioned in her conversation that sometimes it is possible that what you think, the perception about that individual may not be correct, because the way they are doing something, it's just their way of doing it, which looks like they're hiding something, but they may not be revealing everything to you, but they may not be doing it wrong either. So it's a good idea first to talk to them and find out that, okay, I feel like you are doing like this, but can you please explain me that why it is being done like that, so that I'm also clear about it, right? And a good way to approach that problem, rather than just going to the director of that institute and saying, okay, I feel this person is not doing right, and please suspend him, and I feel like I have all seen the things are not working there. So that may not be the best approach. But now I think given this situation, two situations: one, you are senior student, you have to look for a junior student, and second situation, you are junior and you are looking for a senior. In both the situation, what will be your approach of addressing this problem if you think there is some suspicious fraudulent cases happening? We are open for discussion again. I hope I'm audible to all of you, right? Hello?
Yes, sir, you're audible.
I hope I have not scared you too much. Please speak up. These are general context.
So the best way would be to inform in case of the person being junior, finding out about suspicious work going on. It would be like after confirming, after talking to the person, if he's still suspicious, it would be best to report it to the guide, and in other cases, well, of a senior suspecting a junior, it would be best to report it to the guide, or teaching the junior person, and teaching him about what is right and what is wrong.
Yeah, these are, I think, good comments. Of course, if you are a junior and you are not able to understand that what the senior is doing, and you have, but you think there is something suspicious going on. So first, you can, always request the senior that, could you please share your lab notebook? Can I, discuss this experiment with you? Can you explain me how you are doing so that I can also learn this thing? And then if you're not clear and person is not giving you the data, not giving you information, hiding and trying to ignore you, and behaving very rudely, you feel more suspicious about it. Then you are trying to talk to your guide that, okay, something is happening, and can you please check that? But if you are senior, I think your responsibility is much more to really first try to explain the right thing to the junior, and tell him this is how it should be done, because the way you are doing it is not right. I think you are making these mistakes, and this should not be reported. Like, you cannot just show one replicate data like this, make this kind of show the image like this, or you should not make the image look like the sharp blurred and present like that. So in different ways, what you have observation for that junior student, as a friend, as a mentor, you can try to first cordially try to explain them these problems, and try to overcome that. But the student, if they still continue, then you see that, okay, this is kind of a practice, the student going to continue in this manner, then you will go to first talk to your supervisor, and tell them that is what is happening. And anybody else, I think these are good points what Surabhi just mentioned. Anyone wants to add any point? Okay, so given the time which we are getting short, so I think this is probably the appropriate way of moving forward, and you can have variety of situations where you have to think about how to handle this particular problem. And let me now bring back the various possibilities by sharing my slides again. Okay, so there could be different context, different situation. We talked about senior to junior students. Advise him or her that you have observed some misconduct and also inform your advisor. And when you are junior student and you are talking to senior student, of course, you have to be very careful that they should not feel offended. And you should not have any misunderstanding because you may not be competent enough to know that experiment. So again, if you are not satisfied, then you can talk to your advisor and inform him. Or if you also see that, okay, you're a student, but your guide has actually, you ask you to, make an image like this or show something, which was the wrong data, you're not satisfied, then you should first, maybe talk to him that is something, what is your observation. But if he's not, giving you the opportunity to speak and give you opportunity to clarify, then you can inform the head or administration. So these are the different ways of a different level how you should handle this problem. But again, as previously discussed with expert, it's always a good idea to talk to people because many times you will have some perception, but that perception may not be right, and you don't want to just create the problems between and misunderstanding within the group because of your little understanding or misunderstanding of the subject. So good to talk to people, look at their data, look at their the lab notebooks, make sure you are convinced. And if you are still finding that there is some suspicious things are going on in your bag, then you feel free to take the next set of actions. So let's look at another expert's opinion that how you should be safeguarding yourself.
I mean, I think if you're the researcher doing the data, you have to be very good at documenting, writing down exactly what was done and why. Typically, when your paper gets written, the methods section is a couple of pages or even less than that. But I think in the actual conduct of the study, you really have to document step by step what you did, where, and why, so that if there's any questions that come up later about misconduct, you have clear documentation of everything you did. And it'll help. Oftentimes, there's disagreements of how you analyze the data, why you did not follow all the steps that somebody else would have done. And I think the best thing is clear documentation of everything you did.
Well, it's a very important point because many times that for taking the same data file, one could have different ways of analysis, right? And there could be actually 5 different ways of analysis, which given that there is so many softwares coming up, there are so many different approaches are there, so many statistical tests are available. It is possible that your understanding for that subject has led you to choose one type of workflow and one type of analysis, right? While somebody else thinks that the workflow 2 or 3 should have been more appropriate. That's not wrong on your side, as long as you have documented whatever you have done. This is how you have taken the raw data. This is how you have processed. This is what you're reporting, and all that should be well documented in the lab notebook. And that's where I keep emphasizing to all the students who work in my lab or who are part of my classes and courses to maintain good lab notebooks. It's really important. Whatever you do, you please write down those. It will really safeguard you very much. Many times, in fact, most times, you would not like to make a mistake knowingly. You are doing something, and if something goes on without kind of paying your very crucial attention to that detail, it can be picked up from the lab notebook. And if you are reporting what you have done and what is reported in your lab notebook, you are very much safeguarded, right? So that's really important thing. So other point which is to be discussed is the consequence of scientific misconduct. I don't want to talk this point too much again. You have seen it a lot already that people's careers damage, loss of employment, retraction out, which tells about the reasons for these retractions. Variety of problems have happened, authors have been banned for publications, financial penalties have been imposed, and of course in some cases you have seen imprisonment as well as the people committing suicide. So there could be a variety of consequences of these kind of scientific misconduct. So again, let's hear Dr. Olvin for the if research misconduct already happened.
When research misconduct is committed, how does that affect the individual investigator, his colleagues as well as the more general university. I think from the researcher perspective, there's a bunch of things that can happen if actually there's a determination that misconduct has actually occurred. And these may include things like you may have to retract some of your research findings, contact the journals, let them know there was misconduct here and the journals ought to retract it. And that would be very personally embarrassing to the investigator. But there's more serious things that can happen aside from embarrassment. After an investigation, they may decide that there should be a formal reprimand. If it's something very egregious, you may actually get fired from your job. And this is something that if it's in a field where a lot of people know each other, you can be sure they will contact your university and find out the conditions under which you were terminated. And this may actually lead to problems getting another job, especially through the rumor mills. More seriously, I mean, if it's something like the NIH or one of the federal agencies, they may ban you from receiving research dollars for a certain period of time. So it could be problematic for you personally. But it goes beyond the effects of the individual investigator. I mean, I think all the colleagues, everyone you work with, all the people that have been co-authors on your papers, now you'll be brandished as working under environment of misconduct. And even if it was a very isolated incident, there will be general rumors about that all the data and everything else that may have been problematic. And so all your colleagues will get associated with a line of investigation that may be not correct or that was associated with misconduct. Even if they were not directly involved or part of it, you're still associated with that. And it could be for the university, it could be personally embarrassing if these things get out. We know many Ivy League institutions and how they've been severely tarnished when these cases have come up and they've become publicly available. It's the kind of stuff the media loves. It's the kind of stuff the rumor mills love. And it's the kind of thing that brings your institution down. And I could see both the colleagues and the institutions being very upset and reacting to this accordingly.
All right. So that was kind of, just a very good conclusion of various points which we have discussed. So coming to what is the solution for this problem, right? One way, I think, as some of you mentioned, that the research funds could be a problem. So if we have consistent funding for the scientists and the labs, probably that will refrain them for thinking about these activities. Also, giving the improved instructions to the students, researchers, authors, as well as the reviewers, that what is considered to be misconduct, what is considered to be fraudulent data. And I'm hoping that this kind of communication skill course given to you at the very early stage is probably one of the ways to really alarm you, alert you, and give you much more focused approach of being more ethical toward the subject, toward the research, right? So also there are centralized databases for scientific misconduct which people can search. There should be more uniform guidelines for the retractions, and institutions and national agencies should have the Office of Research Integrity, which could investigate this kind of scientific misconduct, offer more courses and tutorials to the students, which I'm sure you will agree that this is really important thing to learn at this stage, so that even unknowingly you should not make any mistake. So finally, let me just conclude with this food for thought, that if you have mistaken that there was a research fraud happening, I think you should try to rectify that. You should try to correct that. You have an obligation to act on this, as per National Academy of Science Code. And let me finish this by showing you this clip, which we talked in case of Dr. Wang Gosok, right? What was the public sentiment for him when he was kind of being investigated? All right, so as you can see, these are large rallies. People are, totally on the road in support of Dr. Wang Gosok. So much public sentiment for, that whether something is going wrong against him and why people are trying to put him this kind of, scandals. So public had so much sentiment and support for him, although it did not turn out to be true at the end. So finally, your reputation of doing this kind of work is most valuable possession. And if it is tarnished, it's very much impossible to restore that. So it's really important that when you are working in the field of engineering, medicine, biology, research field, uh, public is really going to get affected from your findings in one or the other way, right? Whether you make a device, you make a bridge, you make a product, you make a healthcare product, from each one of those individuals and society, they do get positively or negatively affected. So they're quite emotionally attached with your work and with your findings, with your claims, with your success, with your journey. So I think you have an obligation to really fulfill that and try to live up to their expectations and their standards. So it's really, I think, important food for thought. I hope this whole 3 lectures session which I gave you has given you good kind of the overview of not only making good presentations and trying to write originally, also avoiding the various issues of plagiarism and scientific misconduct. We talked about different reasons which are associated with the scientific paper retractions, and of course several ways and issues how we can think about to avoid this kind of scientific misconduct and how to overcome these problems. I hope these 3 lecture sessions were good start for you, where at least giving you some stimulation, some new ideas, new thoughts. Please read more, try to be more original, try to keep making a practice of reading papers, trying to understand them in your own way, and trying to write originally. Also practice for the presentations with the more strategies. And when you come to the research labs, you are doing research experiments, make sure you are following these practices very well. You are documenting all the information very well, and you are avoiding any kind of these issues so that you can come out with the proud of your achievements, and your institution, your family, your country, everybody should feel proud of your accomplishments. Thank you very much for attending these 3 lectures. Now, next instructor, Professor Punekar, will be taking over from tomorrow onwards, and he will talk to you for next week. Thank you very much. If you have any comments, I'm now open for any comments and suggestions from all of you. Any points anybody want to make?
Hello, sir.
Yes.
So I wanted to ask about the assignment that we submitted. It was meant to be submitted over that Google Drive now, not over your mail or something.
So Google Drive I wanted so that everything is in one place. I think some people have faced some issues, so they have also emailed me. But I would prefer if everybody can upload their things in the Google Drive link till tonight by 8 p.m.
Yes.
All right. Any other points to be made? Anything you want me to clarify from these 3 lectures?
Yes, sir. I'm Buddhadev here.
Yes, Buddhadev.
Yeah. So in a scientific experiment, there could be negative results. So what if that the negative results or my mistake in the experiments could be valued or taken as a scientific misconduct?
No, see, I think the main thing is when you are doing the any experiment, you are documenting everything in your lab notebook, and when you are writing the reports based on that work, you are showing that what was your the study design and what the experiments you perform, and to make the conclusions what kind of data sets you have. So of course, every data you are not going to put in the main manuscript, but it is a good idea to put everything in the supplementary data sets and also provide the raw data sheets in your, in the manuscripts. So again, nothing is negative. Of course, you need variety of different type of, the control, different type of other ways to look for your experiment. And even if your interpretation for anything is, without that, you should show those results. And again, everything is not in the main manuscript format, but as long as you are referring the entire data in somewhere in the manuscript, that is, I think, accurate. But in general, there is nothing like a negative result, because it's the observation which we make, that how we performed a study, and few things look like this, other things panned out slightly differently. Now, how we interpret that, so that is actually our way of interpretation for that particular work. I also mentioned to you that same data you take, and it can be processed by 5 different experts in 5 different ways. And there is no one best way of doing that possibility. As long as you're also reporting that your obtained data and the results what you are reporting is done based on this particular type of workflow and what was the rationale of doing that, I think that's pretty much taken care of. So that's where then the reviewers may, if they have any kind of disagreement, that you have to show more of these results, you have to show more of the control data sets, you have to show more of the additional data set, they will come back to you and you are supposed to address those comments and provide more information. So as long as you're not hiding these things, deliberately, you are okay, because you are providing all the data to the community and to the reviewers. So in that case, you should be fine. So nothing like a negative data. It's just matter of how you are interpreting it, and you are still showing the whole data set. Anybody else? Arnab, you still have any comment? Your hand is raised. I know I've exceeded the time now, so it's almost 12:40. If anybody has any specific comment or points to talk, please let me know. Otherwise, I'll stop this particular lecture. And it was kind of a different experience for me to teach as well this time, this course. I must say that probably you can see me. I cannot see any one of you. So it's more virtual experience. I'm hoping that at least 50 people are watching me and they're doing something from these lectures. So it's a different experience, unlike the class when you're really talking eyes to eyes and contacting with the students. But looks like many of your comments are pretty, pretty relevant and very much to the point. So I'm hoping you are overcoming all these online teaching challenges right now, and you're still expressing the complete interest into what the courses you are taking, and you are trying to get the best use of these resources and these kind of ways of learning and communication. So thank you very much for taking this course and being with me for these 3 lectures. I'll be happy to read your assignments and look forward for in-person communication and further communication with you when you come back to IT Bombay campus. Thank you very much.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you so much, sir.
Thank you, everybody.
Thank you, sir. Thank you, sir.
Thank you. Welcome.
Sir.